Hello judges, we are students from Hong Kong Middle School. Today, we are going to introduce about our project, Principle Blocking Drugs for Alzheimer's Disease. Let's look our content. The first part is about production concept. The second one is materials analyzed. The third one is practical rule. And last one is our summary of results. Part 1, Production Concept. For our research motivation, we have two levels. First is social level. Nowadays, the aging of the population in society is getting more and more serious, and diseases that have been accumulated over time are also bringing out during this period. The second motivation is about medical reasons. Alzheimer's disease cannot be effectively cured, so we plan to create a drug to alleviate and prevent Alzheimer's disease by isolating the causative factors, increasing the recovery of nerve cells, and reducing the loss of neurotransmitters. We have three research proposals. First is about we want to make a drug that can protect the elderly in society from Alzheimer's disease in a more real way. The second is our drug not only play a certain role in preventing and relieving Alzheimer's disease. Last one, the ingredients inside can adjust the overall function and hormonal secretion of the body in a balanced state. And then you maybe ask us, what's special about yours? Our drug can alleviate the symptoms of Alzheimer's patients to a certain extent. The advantage is that elderly people who have not yet suffered from Alzheimer's disease can also use this drug to block and protect and repair nerve cells in the principle. Therefore, preventing Alzheimer's disease can also repair cells that have oxidative stress in the body. The second part is experimental materials. These are our material, main substance, amino acid, and vitamins. And all these materials can help our body to do the circulation, or someone is good for our brain. The third part is about practical rule. The first function is to remove harmful substances in the body as other vitamins and anthocyanins will enter the blood circulation and the body's remaining harmful substances are removed, which anthocyanins will ensure that the activity will not be weakened by the elimination of harmful substances and environmental factors. The second process is nucleic acid promotes division, and then ganglioside and vitamin B12 promotes cell membrane repair. The last process is reduces the hydrolysis of acetylcholine, which allows the communication between nerve cells to go more smoothly. And we also have a special work structure called three-stage capsule. The first one is used to wrap aspartic acid, tryptophan, and arginine, and melt them in the stomach. The second one is used to encapsulate glutathione, SOD, anthocyanins, and catechins and melt them in the intestines. The third type is used to include L vitamins and gangliosides, nervinic acid, and heparin A. It takes 2 hours to dissolve in the intestine. This is mainly due to substance in the second and third coatings. If they are combined and advanced before enter the blood and intestinal fluid, not only will the activity be consumed in advance, but a certain degree of rejection will also occur and the efficacy will be reduced. So, special coatings are needed to distinguish them. In addition to drugs, a large amount of starch is used as filler. The last part is summary of results. Let's conclude our innovation point. Our innovation is mainly to carry out multi-factor work. At the same time, we use the combined effects of multiple types of drugs to treat Alzheimer's disease to truly and effectively alleviate it. The places where we start are very central to Alzheimer's disease. At the root of the disease, we remove ROS, repair nerve cells, relieve oxidative stress, replenish the nutrients needed by the brain, and finally increase the exchange rates of neurotransmitters to return the overall brain function to a normal balance point. That's all our introduction. Thanks for listening.